Hi, my name is Shreya, and in this video, I will be explaining how Brain Scanology's shape analysis tool, Genie, precisely quantifies subtypes of sickle cells. The sickle cells analyzed in this video were provided by Dr. William Eaton and Dr. Troy Selmer from the National Institutes of Health. Sickle cell disease is a genetic disease which causes red blood cells to become deformed or sickled. We identified 4 shape subtypes in the sickle cell images provided by Dr. Eaton and Dr. Selmer. These shape subtypes were each identified by a color assigned during the preparation of these cells for analysis. The cells categorized as pink cells were small in size and were very dense. Cells in the blue cell category were star-shaped. Green cells were cells which did not sickle. They were rounded. The last cell shape subtype identified were the red cell shape subtype. These cells were spindle-shaped and appeared to have a smooth sickle shape with just 2 points, each at an end of the cell. Genie uses Brain Scanology's LCPC transform to analyze shapes. To perform the LCPC transform, first, a mask of the outline of the object of interest was created, which in this project was a sickle cell. In step 2, a parallel grid was overlaid onto the outline. The third step was to calculate the distance from the baseline of the parallel grid, shown as a vertical dashed line, to the intersections between the parallel grid and the outline of the object. The fourth step was to linearize the parallel grid system. If one line intersects the outline of the object multiple times, the distances from the baseline of the grid to each intersection along one line of the parallel grid are summed, and a graph of the distances was made. Each line on the parallel grid is shown on the x-axis in the step 4 graph, and the distance is shown on the y-axis. The fifth step was to do a fast Fourier transform on this data to create a frequency plot that is a multi-dimensional representation of the sickle cell shape. The x-axis represents the order of frequency bins, and each bar in this graph represents the value of the magnitude for the corresponding frequency bin. In the sixth step, the frequency plot can be converted to a scalar value that represents the shape. For example, all bars can be summed. When performing the LCPC transform, the orientation of the shape of interest is very important. Our criteria for aligning the sickled red blood cells before performing the LCPC transform were as follows. First, we rotated the image so the longest aspect was vertical. The longest aspect is drawn in green in the sickle cell outline shown here. For rectangular cells, the longest aspect was the longest diagonal through the cell shape. Then, the image was rotated such that the concavity, if present, opened towards the right side. The concavity for the cell shown here is outlined in red. If the cell did not have any concavities, then the flatter side was oriented to face the right. Another important consideration is the scale of the cells. When we applied the LCPC transform to the cell outlines taken directly from images provided by Dr. Eaton and Dr. Selmer, the cells had different sizes and shapes. These shapes were referred to as sickle cells at scale. However, shape is distinct from area. To analyze just the shape of each cell, without considering the size, we resized all cells such that the axis where the difference in size was greatest was scaled to be the same. We constrained the aspect ratio during resizing. These resized shapes were referred to as the pure shapes of sickle cells. In this video, we will be analyzing both sickle cells at scale and the pure shapes of sickle cells. Sickle cells at scale had both different sizes and different shapes. These were the frequency plots created using Genie on sickle cells at scale for each of the 4 sickle cell shape subtypes. Comparing these 4 plots, the height of bin 3 relative to bin 2 changes between cell shape subtypes. For all 4 frequency plots, bin 2 is boxed in pink, and bin 3 is boxed in blue. An index can be made to distinguish between sickle cell shape subtypes based on this observation. The index used here was the ratio of bin 3 over bin 2. Box plots depicting the ratio of bin 3 over bin 2 are shown here for each sickle cell shape subtype. Pink cells had the lowest median ratio and red cells had the highest median ratio. T-tests of the ratio of bin 3 over bin 2 between all combinations of two sickle cell shape subtypes demonstrated that the ratio of bin 3 over bin 2 is different. Therefore, this index demonstrates that the step pattern observed in bins 2 and 3 of the frequency plots can distinguish between sickle cell shape subtypes for real sickle cells measured at scale using Shape Genie. We then analyzed the pure shapes of sickle cells. To develop the pure shapes, the sickle cell shape outlines were resized such that they all had the same height, while constraining the aspect ratio. These were the frequency plots created by using Shape Genie on the pure shapes of sickle cells for each of the 4 sickle cell shape subtypes. Looking more closely at the first 10 bins, there are differences in the height of bin 3 relative to bin 2 between the 4 shape subtypes for the pure shape sickle cells. For each frequency plot, bin 2 is boxed in pink and bin 3 is boxed in blue. There are clear differences in the step patterns between bins 2 and 3 for each of the 4 sickle cell shape subtypes. Correspondingly, an index was created based on this observation. The box plots shown here depict the ratio of bin 2 over bin 3 for all sickle cells within each of the 4 sickle cell shape subtypes. For the pink cells, a large Y outlier was removed from the box plot to shorten the Y-axis and spread out the box and whisker plots. T-tests on the ratio of bin 2 over bin 3 between all combinations of 2 sickle cell shape subtypes demonstrated a significant difference in this ratio between the shape subtypes. Therefore, the step pattern can be used to derive a robust index that stratifies sickle cell shape subtypes when measuring pure shape of sickle cells. In conclusion, Shape Genie can objectively quantify differences in sickle cell shapes in ways that the human eye cannot. Shape Genie quantifies shapes more robustly than traditional approaches such as area. Potentially, Shape Genie can allow for the discovery of new morphological subtypes, which can lead to more specific research focuses and potential therapies for sickle cell disease. Some potential applications of Shape Genie include precisely quantifying how therapies alter sickle cell morphology within each cell shape subtype. Shape Genie could also be used to quantify how morphological changes affect clotting propensity and resistance to declotting treatments. Our results can lead to the development of better machine learning models to classify the 4 shape subtypes of sickle cells that we have identified. Finally, Shape Genie could be applied to calculate more precisely how therapies affect the percent enrichment of morphological subtypes. Thank you for watching this video.